Everyone, in this video we're focusing on metabolism. So in a nutshell, we're going to describe the most important chemical reactions in our body that produce ATP, and then the most important reactions that store energy for later use. So we're going to frame this video around two main concepts that are basically the opposite of one another. The first one is catabolism. And this is a word that describes all the reactions that produce ATP. And the second one is anabolism. And this is a word that describes all the chemical reactions that use ATP in our body. So catabolism produces ATP, anabolism uses ATP. And then within these categories, we're going to focus on the two main macronutrients that are involved in giving us ATP and storing energy. The first one is carbohydrate, and the second one is lipid. So we're going to talk about carbohydrate catabolism, carbohydrate anabolism. Same thing for lipid. Lipid catabolism first, then lipid anabolism after. So this is a slide that we've already seen in a previous video, but it's to get these concepts back fresh in our memory. Remember that based on the first law of thermodynamics, energy cannot be created or it cannot be lost, but it can be converted from one form to the other using chemical reactions. And we already described two types of chemical reactions. On the left-hand side here, we had endergonic reactions, and these are reactions that increase the potential energy. In this type of reaction, our body uses ATP in order to create chemical bonds, and these bonds have lots of potential energy, right? So that's when we store energy for later use. In contrast, we had exergonic reactions where the potential energy of the products is lower than that of the reactant, right? We decrease the potential energy level during an exergonic reaction. But what we get instead in an exergonic reaction is the production of ATP. And remember that ATP is the energy currency of the cell, right? So when we have ATP, we can use this to power all the work that we want. Our muscle contraction, our heart beating, us breathing air in and out, our sodium and potassium pump, and so on. So we already have these two categories defined in our mind. Now we're going to add the word catabolism and anabolism to that. So as I mentioned in the introduction, all reactions that produce ATP are called catabolic reactions. So catabolism goes with our exergonic reactions, while reactions that use ATP will fall under the anabolism umbrella. So very similar concepts, just adding another kind of piece of vocabulary on top of the concepts. And this is a flow chart that hopefully will help you put the accurate words to define and describe catabolism and anabolism. So as we already mentioned, catabolisms are reactions that will break down large biomolecules and take the potential energy that was stored in the bonds of those large biomolecules to release energy. As a result, the potential energy of the products will be lower than that of the reactant, and use that to release energy. Another word of saying that is it releases ATP, as we can see at the bottom there. The other arm of metabolism is called anabolism. And as we know, these are all the reactions that synthesize large biomolecules. But in order to do that, it requires an input of energy. Or you can say that it requires an input of ATP. So that at the end of an anabolic reaction, the potential energy of the products is higher than that of the reactant. So we already discussed these questions, but these should be the type of questions that you ask yourself when you're studying. So are catabolic reaction exergonic or endergonic? Catabolic reactions, because they produce ATP and they lower the potential energy, are exergonic. What about anabolic reactions? Other way around. Anabolic reactions are endergonic. So the three main concepts here are exergonic versus endergonic, catabolism versus anabolism, and how this relates to ATP, either its production or its use. Now let's put this back in context of our everyday life. So as we go along our everyday life, both catabolic and anabolic reactions occur at the same time in our bodies. But what's important is the balance. At any one time, it might not be 50% of anabolic reaction and 50% of catabolic reaction. It might be 60-40, or it might be 70-30 or 80-20, right? So it's the balance that's really important. And the balance is determined by what our body's needs at any one time. So my question to you is, when do catabolic reactions dominate? When do we have more catabolic reactions occurring compared to anabolism? Another way of asking this is, when do our need from ATP increases? When do we need a lot of ATP in our life? Let's look at the four situations at the bottom. Number one, when you're exercising, there's a lot of work that you need to do. You need to contract your muscle, your heart needs to beat faster, you need to breathe faster and deeper as well. So all of these processes require ATP. So exercising is probably one of the most typical examples of where catabolism dominates in our metabolism. Moving on to number two, growing. So when you grow, you are developing new tissues, right? You need to build more muscle. You need to build more bones. You need to build more fat, and so on. So it's really a time where we need to build more of those large biomolecules. So in growing processes, we use ATP, and then we take that ATP and convert that energy into a lipid, into a protein that can be used for a muscle, into a carbohydrate that we can store in the liver, for instance. So this is an example where anabolism would predominate. Number three, fasting. So intermittent fasting is getting more and more popular. People fast for Ramadan, for instance. So let's think about what happens, for instance, at the end of a 12-hour fast where you haven't eaten for 12 hours. So at that time, you still need ATP to power, the basic processes of your cells. But you cannot get that energy from the food that you've eaten because you've been fasting. So what does our body do? It's going to go into our stores. So our stores are carbohydrates and our stores of lipids in our body. And it's going to start breaking down those chemical bonds in order to produce the ATP that we need. So that's the definition of catabolism. Finally, number four, being stressed out. So under situations of stress, there's a lot of processes in our body that are increased. Your heart rate's going to increase, your breathing rate's going to increase, you're going to become hyper aware, so your brain's going to need more ATP as well. So all of this requires an increased need for ATP. So being stressed out is also an example where catabolism would predominate in our body. I hope this helps you understand how these concepts fit into our everyday life. So now we're really zoning into catabolism. Okay. So the role of catabolic reactions, as you know now, is to produce ATP. The question here is, how do we get there? So this is a flow chart that summarizes how we go from large carbohydrates, so either from our food, like the grains that we eat or the fruit that we eat, or from carbohydrates stored in our body in the form of glycogen. So how do we get from that all the way to ATP? So this is a summary. This is an introduction slide, and we're going to go into more details in the next couple slides. But let me walk you through some of the key concepts here and the key principles here. So let's start at the top here. Okay, we get carbohydrates in our body mainly in one of two forms. So if you just ate a big plate of pasta that has lots of carbohydrate, you get this influx of glucose. So we can get glucose from our food. Now our body isn't really good at storing glucose as a one molecule of glucose. So the other way that we find carbohydrates in our body is in the form of glycogen. So what we're seeing here is this representation of glycogen. But what I want you to think about, I want you to think about glycogen as a tree of glucose. So the center here, there's a core, like the trunk of a tree, that is a protein. But then from this trunk, you can see that it has grown all of these branches. And on the branches, every leaf is a molecule of glucose. So each of the little dots that you see here is one molecule of glucose. So in one molecule of glycogen, you can have 20,000, 30,000 molecules of glucose. So what happens when we need to produce ATP from carbohydrates, we can either start with glycogen and start peeling off those glucose leaves. So glycogen can be converted back into glucose. And then glucose is converted to pyruvate. Pyruvate is converted to acetyl-CoA. And then acetyl-CoA can enter the citric acid cycle, which produces lots of electron carriers. We'll see NADH and FADH2. And at the end of the day, those electron carriers enter the electron transport chain, whose main goal is to produce ATP. So you can think about this as this like cascade of chemical reactions. Now, each of those steps actually contain like 8, maybe 10 specific chemical reactions. For the purpose of this course, we don't need to know all of the steps, but we need to know the names and the function of these overall reactions. So starting here, the conversion of glycogen to glucose is called glycogenolysis. So glycogenolysis. Step 2 is the conversion of glucose to pyruvate, and this is called glycolysis. Step 3 is the conversion of pyruvate to acetyl-CoA, and this is called pyruvate oxidation. Then, once we have acetyl-CoA, it enters the citric acid cycle, produces NADH and FADH2, which finally feeds into the electron transport chain in order to produce ATP. So we have 3 main catabolic reactions on the carbohydrate side: glycogenolysis, glycolysis, and pyruvate oxidation. Now there's one thing I want to bring to your attention, and it's the end of the words. We have glycogenolysis and we have glycolysis. Okay, so they end in the same way because lysis means the breakdown of. So when you're trying to learn these terms, you don't really need to memorize them. You just need to look at the word. If you know that lysis means breakdown, then you know that glycogenolysis. So it's a breakdown of glycogen into glucose. Same thing for glycolysis here. Glyco refers to glucose and lysis is the breakdown. So glycolysis is the breakdown of glucose to pyruvate. Now moving on to lipids. So first, lipids is just a fancy word for fat. So there's a wide variety of the fats that we eat and we store in our body. But the main molecule of fat that we use for ATP production is called triglyceride. And you see the triglyceride here. So I use the analogy of an alien with 3 legs, where the triglyceride here in red has a head of a glycerol, and then it has 3 fatty acid legs that I'm highlighting here in purple. So the first step in order to make ATP from lipids is we need to break down this alien with 3 legs. We need to break down this triglyceride into its individual component. And that's what we see here. The first step is breaking down triglyceride into 1 glycerol and 3 fatty acid. This reaction is called lipolysis. Once again, notice how it ends in lysis. So when you see lipolysis, lipo for lipid, lysis for breakdown, you know it's the breakdown of lipids. More specifically, it's the breakdown of triglycerides. Now at this point, the glycerol will go one way and the fatty acids will go another way. So the glycerol here can actually enter glycolysis. So we'll just enter glycolysis and then follow the same pathway that we've already described for carbohydrate. The fatty acids are then converted to acetyl-CoA. And this is called beta oxidation. Once again, once we have the fatty acids converted to acetyl-CoA, they can follow the same pathway that we've talked with carbohydrates. So first into the citric acid cycle, and second into the electron transport chain. And that's how they produce ATP. So here's a neater version of this same information. And this is the framework that we're working with for the next 30 minutes or so of this video. So we're starting with carbohydrates. So now we're going more in detail into each of the steps of carbohydrates catabolism. So on the left-hand side here, we see the breakdown of glucose, and you'll recognize some of the key molecules that we already talked about. We start with glucose into pyruvate. That's step 1. That's your glycolysis that we just described. Then from pyruvate to acetyl-CoA, that's our pyruvate oxidation, the citric acid cycle, and the electron transport chain. So it's the same cascade that we just described, just in a different way. So as I said, we don't need to know the details of all of the 9 steps, 9 chemical reactions that happen between glucose and pyruvate. What we're focusing in for the purpose of this course, we need to know the name of the reaction. We need to know how it starts with glucose, how it ends with pyruvate. And we need to know the energy yield. So how much energy are we actually producing through this pathway? So when we talk about energy yield, we'll focus on basically 3 main molecules. ATP, for obvious reason, because it's energy. And then our electron carriers. Our electron carriers are NADH and FADH2. So step 1, as we described, is glycolysis. It starts with glucose, ends with pyruvate. And for 1 molecule of glucose, this pathway creates 2 molecules of ATP and 2 molecules of NADH. Importantly, you can see in this cartoon that the glycolysis here all happens in this yellowish part, and that's meant to represent the cytosol of the cell. And that's in contrast with everything else below that, which happens in the mitochondria of the cell. So glycolysis is the only thing that happens in the cytosol. The other unique thing about glycolysis is that this part does not require oxygen. Another way of saying this is that it's an anaerobic reaction. We'll see that everything else below, everything that happens in the mitochondria, needs oxygen in order to keep going. Step 2 is our pyruvate oxidation. Right? It's when we take pyruvate and then we make it into acetyl-CoA. The energy yield, once again, from 1 molecule of glucose is 2 NADH. And as we said, this is a reaction that requires oxygen, or we call this an aerobic reaction. Step 3 is our citric acid cycle. So as the name says, it's a cycle. So it starts with acetyl-CoA and it's going to run through a series of reactions. And at the end, what we're producing is CO2. For 1 molecule of glucose, we produce 6 NADH, 2 FADH2. So these are electron carriers, and then 2 molecules of ATP. The 4th and final step is your electron transport chain. So we'll see more details later, but basically what happens is that the electron transport chain takes all of the NADH that were produced higher up, so the NADH produced from glycolysis, the NADH produced from pyruvate oxidation, and the NADH and FADH2 produced from the citric acid cycle, and it turns them into ATP, a lot of ATP. And the byproduct of that, the end product of the electron transport chain, is actually water. So if you think of the energy yield from the top to all the way to the bottom, we get 2 ATP from glycolysis, we get 2 ATP from the citric acid cycle, and then we get 26 to 28 ATP from the electron transport chain. When we add them all up, for 1 molecule of glucose that goes through the entire process, we can get 30 to 32 molecules of ATP, which is quite a bit of ATP when you think about it. So in the next slide, we are zooming in into the glycolysis part. Okay, so we're just taking glucose to pyruvate and looking at it a little bit more in detail. So on the left-hand side, the cartoon represents glycolysis. We recognize it because it starts with glucose and it ends with pyruvate. Now, as I said, here we see all of the 9 steps of glycolysis. We don't need to know all of these intermediates or all of these steps. But I want to highlight a few things. So glycolysis is basically a 2-step process. The first part at the top here, I want to bring your attention to the fact that we need to use ATP in order for this reaction to happen. So the 2 steps that I've highlighted in blue here, you can see how ATP is used and converted to ADP for some of these steps to happen. So the early steps in glycolysis require the use of ATP. Would that make this part of glycolysis endergonic or exergonic? Because we use ATP, this part is actually an endergonic part of glycolysis. And we lose 2 ATP in the first couple steps of glycolysis. But we gain them back in the later steps of glycolysis. So the first thing that's important to notice here is that anything below that red line will happen twice for 1 molecule of glucose. That's why you see like the 2 here in red, is to show you that everything happens twice below that line. So now let's look at what is produced during those later steps. So you have 1 molecule of ATP produced here, you have another molecule of ATP produced at the bottom here, and then you have 1 molecule of NADH produced there as well. So because we're producing ATP here, these would be exergonic reaction. And the energy yield from that is 2 times 2 ATP. So 1, 2, 3, 4. This is where our 4 ATP is coming from. And then 2 times the NADH. So finally, when you look at the overall glycolysis, we've invested 2 molecules of ATP at the start, so minus 2 ATP. But then we've made up 4 ATP and 2 NADH. So total amount of energy that we get from glycolysis is 2 ATP and 2 NADH, which exactly fits what we had in our previous slide. So hopefully that helps you understand a little bit more where the ATP and the NADH are coming from in glycolysis. Our second step was our pyruvate oxidation. And we have to think of pyruvate oxidation as the branch point, like the deciding point, whether we're going to go down the aerobic pathway, which is our citric acid cycle and our electron transport chain, or the anaerobic metabolism. So let's start with the right side here, because it represents what happens most of the time. So when we're operating in an environment where there's plenty of oxygen, your pyruvate will be converted to acetyl-CoA via pyruvate oxidation, And this acetyl-CoA enters the citric acid cycle and the electron transport chain. This is the same pathway that we described a couple slides earlier. And we know that through this, we can yield 30 to 32 molecules of ATP. However, there are situations in our life where oxygen is limiting. So you can think, for instance, of living or hiking at high altitude where oxygen is rarer than it is at sea level, like here in Halifax. Or you can think of like exercising at high intensity where the need for oxygen is actually greater than what our body can use. So these are examples where oxygen becomes limiting. At this point, pyruvate can no longer be converted to acetyl-CoA, but it can be converted, and it is converted to lactate. And that's what you can see here. So you have pyruvate being converted to lactate, and that's the anaerobic branch of the glycolysis. To do that, we actually lose 2 of the NADH that we produced. So the anaerobic conversion of glucose to pyruvate then to lactate, the energy yield is only 2 ATP. And that came from the glycolysis, what we just described, right? So I want us to kind of pause for a second here and compare the energy yield of the aerobic pathway, 30 to 32 molecules of ATP per molecule of glucose, to the anaerobic pathway, where we only have 2 molecules of ATP per molecules of glucose. So that's one of the reasons, for instance, where when you exercise at really high intensity and you're relying on your anaerobic pathway, you cannot sustain that for a very long time because you're only producing much lower amount of energy than you were if you were relying on your aerobic pathways. Step 3 is our citric acid cycle. The main goal of the citric acid cycle is to produce electron carriers. So once again, there's many intermediates in that cycle, but I want us to focus on the energy yield of that cycle. So we start with acetyl-CoA and it enters the citric acid cycle. And then let's highlight the 3 NADH that are produced through that cycle. So we have 3 NADH. We're going to have 1 FADH2. So these are our electron carriers. And then we have 1 molecule of ATP right here. So what I just wrote here is the energy yield for 1 cycle of the citric acid cycle. Okay, so that's going around once. Now for each molecule of glucose, we actually get this cycle going on twice. So when we talk about the energy yield, we always talk about how much energy you get for 1 molecule of glucose. So in this case, we have to multiply all of these values by 2. So the net energy yield for 1 molecule of glucose is 6 NADH, 2 FADH2, and 2 ATP. So we're still not producing a lot of ATP, which is really what we want at the end, but we are producing a very significant amount of these electron carriers. And then our 4th and final step is the electron transport chain. The main goal of the electron transport chain is to convert all of the NADH and all of the FADH2 that we produce higher up in the pathway, so in the glycolysis, pyruvate oxidation, and the citric acid cycle, and converting those into ATP. So let's first look at the cartoon and look at what's happening here. So in the bottom here in yellow is the cytosol, right? That's where glycolysis happens. And in blue here, we're into the mitochondria. That's where the citric acid cycle takes place. Now the mitochondria is an organelle that has 2 membranes. So it has the outer membrane right here, and it has the inner membrane right there. And you can see that it's in the inner membrane that you have a series of different proteins. And the role of these proteins is to take all of these electrons, all from NADH and FADH2, and create a proton gradient. Okay, so create a concentration gradient of hydrogen ions, or H+. So you can see here that in between the two membranes, we have lots and lots and lots of H+. So we have a situation where we have high concentration of hydrogen ions in that space that we call the intermembrane space. In contrast, in the mitochondria, we have a low concentration of hydrogen ions. What I'm trying to highlight here is that there's a concentration gradient where we have lots of hydrogen inside the intermembrane space and little hydrogen into the mitochondrial matrix. So we have a concentration gradient. And this concentration gradient is so important because it is the energy used to power the last piece of the electron transport chain, which is the ATP synthase, which is right here. So the ATP synthase is an enzyme, as the name says, that synthesizes ATP, okay, that makes ATP, that produces ATP. So you can think of this energy conversion as basically a two-step process. First, we have energy in the electrons of FADH2 and NADH, and this energy is converted first into a concentration gradient of hydrogen ions. So that's energy conversion number one. Energy conversion number one. And then the ATP synthase takes this concentration gradient and converts this concentration gradient into ATP. So that's energy conversion number two. So basically, through research, we've shown that you can create 2.5 molecules of ATP per molecule of NADH that reaches the electron transport chain. And you can create 1.5 molecule of ATP per molecule of FADH2 that reaches the electron transport chain. So I'll spare you the calculation, but that's where you get 26 to 28 molecule of ATP per molecule of glucose. So these are our 4 steps where we take first glucose, and through a series of reactions, we can create energy. We can create ATP from breaking down that glucose through a series of catabolic reactions. So now let's recap each of those reactions and how much energy is yielded through each of the steps. And hopefully that helps you, give you a summary and then make things a little clearer and straighter in your head. So remember that the things that we need to know for each reaction are the name, how it starts, so what's the reactant, how it ends, what's the product, and the energy yield in terms of NADH, FADH2, and ATP. So step 1 is glycolysis, where we take glucose and convert it into pyruvate. During glycolysis, we produce 2 NADH. We do not produce any FADH2, and we produce 2 ATP. Remember that these 2 molecules of ATP come from the fact that we use 2 ATP first, but then we produce 4 of them later. So minus 2 plus 4 is equal to 2. Step 2 with oxygen is when we convert pyruvate to acetyl-CoA. We call this pyruvate oxidation. And during pyruvate oxidation, we produce 2 NADH, but no FADH2 and no ATP either. Now the other route that pyruvate can take is be converted to lactate if oxygen is limiting. We call that pyruvate reduction. During that reaction, we would actually use 2 NADH, so we would lose those NADH, and we'd produce no ATP and no FADH2. During the citric acid cycle, we're converting the acetyl-CoA to carbon dioxide. As we know, the main goal of the citric acid cycle is to produce these high energy electrons. So we're going to have lots of NADH, 6. We're going to have some FADH2, 2, and then we're also producing 2 ATP. And finally, we take all of the electron carriers that we've produced and we use them in the electron transport chain. So the 10 NADH that we've produced here are being used, so we lose them, but that's okay because we're converting them into ATP. We're converting our 3DH2 as well, and that produces 26 to 28 molecules of ATP per molecule of glucose. So here's a neater version of that same chart, and I hope this helps kind of summarize and understand where that energy is coming from through this series of catabolic reactions. Now, the one thing that we haven't mentioned with regard to carbohydrate is what happens when we start with glycogen instead of starting with glucose. So we already know that the breakdown of glycogen is called glycogenolysis. So now when we break down glycogen, it actually breaks down into a cousin of glucose, which we call glucose 6-phosphate. But you can think about it as basically having the same function as glucose. This glucose 6-phosphate will enter glycolysis right here at the top, and we'll be able to produce the same NADH and the same ATP that we would have had with the glycolysis that we just described. So what we just did in the last couple of slides is really zone in to everything carbohydrate related, right? From glycogen to glucose to pyruvate to acetyl-CoA to the citric acid cycle and the electron transport chain. In the next couple of slides, we're looking at the lipid side of things. So how do we break down triglycerides into glycerol and fatty acids, and then fatty acids into acetyl-CoA? So here we have the 4 steps. You'll first notice that the last 2 steps are the same, right? So we still want our lipids to enter the citric acid cycle and then the electron transport chain. It's the first steps that are going to be different between carbohydrates and lipids. So first, let's remind ourselves that the main lipids that we store in our body that we use for ATP production are triglycerides. And triglycerides can be thought of as an alien with 3 legs, right? Where the head here is the glycerol, and then each of the 3 legs are individual fatty acids. So the first step is breaking down triglyceride into 1 glycerol plus 3 fatty acids. So we are basically cutting down the chemical links between the glycerol and each of the fatty acids. And that's called lipolysis. Now step 2 is divided into 2. Your glycerol goes into glycolysis and your fatty acid goes into beta oxidation. So beta oxidation is taking fatty acids and converting them into acetyl-CoA. Once that's done, we know acetyl-CoA can enter the citric acid cycle and produce all of these electron carriers that we already talked about. Same thing, within glycolysis, glycerol will be converted to pyruvate, which can then be converted to acetyl-CoA and then follow the citric acid cycle and the electron transport chain. And that's exactly what you can see in the cartoon here. We start with a triglyceride, and then step 1 is lipolysis, where we're creating glycerol and fatty acid. Glycerol enters glycolysis. Fatty acids, via beta oxidation, create acetyl-CoA, and that enters the citric acid cycle. So it's the same information on both sides, just laid out differently. And here is a neater version of that same information once again. Now, the difference between lipids and carbohydrates is that with lipids, I cannot give you a specific number of ATP produced per lipid because we really have a number of different lipids that are possible in our body. So we have lipids that are very, very short. that make relatively few molecules of ATP, but then we have lipids that are really long chains of carbons, and those have the possibility of making lots and lots of ATP. So I cannot give you a specific number, but in general, the number of ATP from lipids that we can produce is much greater than what we can produce from carbohydrates. And that's just because lipids are typically bigger molecules, so they have more of those bonds, therefore more potential energy. So this ends our discussion on catabolism. But we have this whole other arm that we haven't explored yet, right? And that's anabolism. The way that I want us to think about anabolism is basically as a mirror image of the catabolic reactions that we just talked about. So these are all reversible reactions. For each catabolic reaction, there is the opposite anabolic reaction. So for instance, glycolysis was the breakdown of glucose to pyruvate. Well, we have this reaction that can take pyruvate or lactate or amino acid and convert it back to glucose. So it's the opposite of glycolysis, and it's called gluconeogenesis. Same thing with glycogen. So the breakdown of glycogen to glucose 6-phosphate, we call this glycogenolysis. The anabolic equivalent is taking glucose 6-phosphate and putting that into glycogen, and that's called glycogenesis. And finally, on the lipid side, we had the combination of lipolysis and beta oxidation, which took triglyceride to fatty acid and then to acetyl-CoA. The opposite anabolic reaction is taking acetyl-CoA, making fatty acids from them, and then building triglycerides. And this is called de novo lipogenesis. Before we go into some of the details here, I want to bring your attention to how the words end on the anabolic side. Notice genesis, gluconeogenesis, glycogenesis, de novo lipogenesis. So genesis means the creation of. So glucogenesis is the creation of glucose. Glycogenesis, here stands for glycogen, is the creation of glycogen. Lipogenesis is the creation of lipids. So that's obviously in contrast with all the lysis words that we had on the catabolic side, which means the breakdown of. So hopefully this helps us organize our thought as to which reactions are catabolic and which reactions are anabolic. So let's look at each of the three anabolic reactions that we just talked about. So gluconeogenesis, which we often abbreviate as GNG, is the creation of glucose from non-carbohydrate sources. So we take something that isn't a carbohydrate, like pyruvate, lactate, or amino acid, and then we reverse glycolysis, so that's gluconeogenesis, and then we make glucose at the end. So this happens primarily in the liver, but it is a very, very important reaction, basically for our survival. So most cells in our body, if you take your heart cells or your muscle cells, they can use either glucose or lipids to make ATP, right? They'll use whatever is there. They don't have a strong preference. There's one exception, and that is our brain cells. Our brain cells have these very, very strong preference for glucose in order to make ATP. This means that if for some reason glucose in our body is limited, that happens in prolonged fasting, for instance, then without this gluconeogenesis, we would be in danger of, starting to have neural symptoms. Now, the best case scenario is a headache, but the worst case scenario are like seizures, coma, or even death. because our brain needs glucose in order to function. So if you don't have glucose, you're compromising your brain function, which literally controls the rest of our body. So think of this gluconeogenesis as this emergency mechanism where if the blood glucose goes low and our brain is in danger of lacking that source of energy to make ATP, then our liver is going to make that glucose from pyruvate, lactate, and amino acid so that our brain can keep functioning. So I cannot understate how important gluconeogenesis is, not only to our health and our well-being, but literally to our survival. And that's an anabolic reaction. So it uses quite a bit of ATP and NADH in order to make that glucose, but it's a trade-off that makes sense given that it's going to allow us to survive. I'll take that trade-off any day of the year. Glycogenesis is the creation of glycogen from glucose. So the actual reaction is taking glucose into glucose 6-phosphate and into glycogen. And you can see that in the cartoon here, where you have glucose conversion to glucose 6-phosphate. And we see here that we're using ATP. And then glucose 6-phosphate is integrated into glycogen via the enzyme glycogen synthase. So this happens mostly in our skeletal muscle and in our liver. And the benefit of this reaction is that our body isn't good at storing glucose as this one molecule of glucose. We can only store a very limited amount of glucose per se. But when you put glucose into glycogen, then it becomes much more easier to store that glucose. So putting glucose into glycogen is basically a very efficient way of building that potential energy so that we can use it later. And the last one is de novo lipogenesis, and that's the creation of triglyceride from acetyl-CoA. As we said, it's a 2-step process. So we first start with acetyl-CoA and make fatty acids. And then we take those fatty acids, combine them with glycerol, and we form triglyceride. And this is what we see in the cartoon here. Acetyl-CoA makes fatty acid. Glycerol comes from glycolysis. And then you take 1 glycerol and 3 fatty acids, put them together, and you have this triglyceride. So now why is this reaction important? Is because our body has this almost unlimited ability to store lipids. So I've just told you how the body is better at storing glycogen than glucose. Well, the body is much, much, much better at storing lipids compared to glycogen. We basically have a limit as to how much glycogen we can store in our muscle and our liver. There's almost no limit as to how much lipids you can store in your body. So most of these lipids go into adipose tissue. So that's your fat tissue, right? That's the kind of fat tissue that nobody likes. But from a metabolic perspective, it makes a lot of sense. So these are also anabolic reactions. We're going to use ATP and NADH to power de novo lipogenesis, and the exact amounts will depend on which lipid is synthesized and how long these chains of fatty acids are.